Bismillahirrahmanirrahim. We start paper two as requested by many of you. I will do a paper four and a paper two so that I can give opportunity to both the AS and the A2 students to be able to figure out these papers and do well in the forthcoming exams. As you all know, the paper two is remained the same. There's not much change in it. There's no timing change. The timing is still 1 hour and 15 minutes, and it's going to be on the same day as the paper five. So you're going to do the paper two and paper five on the same day. It's usually in the timetable for Cambridge exams, it's on the same day. So if you're doing the AS, you'll only do the paper two. If you're doing the composite, you'll do both of them. If you've taken the AS, then you'll only do the paper five that day. So this is 1 hour 15 minutes, and it is how many marks? It's 60 marks. So you have 60 marks to work about. Now let's look at the grade boundary. As you can see, for the June 2022, the grade boundary is the 60 marks, and it is anything between 39, 32, 38 is for the A. And then for the B, it is 34, 27, and 32. And then for the C, it is 29, 23, 26, and then so on and so forth. You've got the D's are 22, 18, 21, and 16, 14, 14. But in Pakistan, we do component 20, 22. We do the 22 component, sorry. So the 22 one will be held in Pakistan. Of course, for different zones, it's different components which are taken. So I'm going to discuss all the three components so that you all can learn from each of them something new. Now, starting with question number one, figure 1.1 and figure 1.2 are photomicrographs showing the distribution of tissue in the lungs. Figure 1.1 is a photomicrograph section through part of the lungs. And 1.2 is a high power view of the area indicated on figure 1.1. So we've got 1.1 and then we've got an enlarged part of figure 1.2. So you can see these two diagrams. So this one first, which is figure 1.1, and 1.2 is this area which has been further magnified. Now let's look at the question. State the names. State the names of the tissues A, B, and D. Let's go back. A, B, and D. What is A? A is the ciliated epithelium because this is the lumen. This is the distribution of tissue in the lungs. So you're going to get some portions of the air passages. So this A would be the ciliated epithelium. B, you can see, is the smooth muscle, which is lining the trachea and the bronchiole, smooth muscle. And D is the plates of cartilage. There is this bit of the cartilage, right? So ciliated epithelium, smooth muscle, and cartilage, and that gets you your 3 out of 3. Then the B part of the question, describe the role of the glands labeled C in figure 1.1 in maintaining the health of the gas exchange system. Now let's look at the glands labeled C. Now this is the glands labeled C. Now what are these glands? These are mucus glands. They produce mucus, and the mucus forms a layer over the ciliated epithelium. And this mucus traps or the bacteria stick to this, or bacteria or virus or microorganisms stick to it, and then the mucus is moved away from the alveoli or the lung tissue and towards back of the mouth, and the mucus prevents the pathogens reaching the epithelial cells or the alveoli or the gas exchange surface. So how do we word all this? Produce mucus, 1 mark. Mucus forms a layer over the ciliated epithelium. 2nd mark, traps the bacteria, or you can say virus, or you can say microorganisms, or you can say pathogens, even you can say dust, pollen, spores. So all those things are actually trapped by the mucus. And then the mucus is moved away from the lung tissue or the alveoli, is taken to the back of the throat by the cilia. And what does this? How does this help? This helps in preventing pathogens from reaching the alveoli and causing severe lung infections, bacterial or viral, or even causing some reactions, allergic reactions from the dust and the spores. The C part of the question, hyperventilation occurs when a person breathes too fast or too deeply. The effects of hyperventilation are a decrease in partial pressure of carbon dioxide in the alveolar air and an increase in the pH of the blood. Figure 1.3 shows the changes in the oxygen dissociation curve as a result of hyperventilation. Percentage saturation of hemoglobin with oxygen, partial pressure of oxygen, and one is at pH 7.4, which is this one. pH 7.4 is this one, right? And then we have another one which is pH 7.7, and that is this one. Now, what are they asking you? They're asking you state the percentage saturation of hemoglobin at a partial pressure of oxygen of 4.0 kPa. So all you had to do was what? Draw a line on 4. So what do we have? So they have asked you all these points. So for 7.7, what do you have? You have nearly 7.2, 72%, not 7.2, sorry, 72% because it's percentage saturation. So that was 72%, and then at pH 7.4, at pH 7.4, what do we have? We have it here at 60%. So you had to just figure that out. That wasn't very difficult. This would be, so this would be 72%, and this would be, I think there's some sort of error in this because they're not giving you a space to write. They've written kPa, 72% and 60%. Part 2 of the question, use the information in figure 1.3 to state and explain the effect of hyperventilation on the supply of oxygen to the respiring tissues. So let's go back to the graph. What has hyperventilation done? You see, they told you something, an increase in the pH of blood. So increase means 7.7. So what has happened to the pH? There's a decrease in the supply of oxygen to the tissues. Now, why am I saying that? You see, what has happened? Previously, it was 60%. Now it is 72% saturated. The oxygen is percentage saturation is now 72%. That means it's not giving up its oxygen. So what does that mean? It means hemoglobin is more saturated. More saturated means it's not giving up. So at lower pCO2, when the carbon dioxide is lesser, there's increased pH of blood. The affinity of hemoglobin for oxygen is higher. Or you can say hemoglobin is more saturated. And you can give me a data quote for difference in saturation of hemoglobin in the correct range of partial pressure of oxygen within 2 to 6 kPa to O2. So basically for 3 marks, you had to just explain the graph to me, that what is happening in the graph. So the fact that previously it was 60% saturated, now it is 72% saturated. So that means the hemoglobin oxygen affinity has increased. So the hemoglobin is not giving up its oxygen. Its friendship with hemoglobin is now more profound. It's not giving up its oxygen. Affinity is a word for friendship or love. That is the word affinity means. So at a lower partial pressure of carbon dioxide, increased pH of blood, the affinity of hemoglobin for oxygen is higher, or you can say hemoglobin is more saturated. And there is, of course, that means there is a decrease in the supply of oxygen to the tissues. So you can give me a data quote as well. Data quote means anything at 2 kPaO2 or between 2 and 6, you could give me a data quote. And as you can see here, I've drawn the lines and I have shown you how this would show here. So you give me a data quote and you got another mark for that. So there were only 2 marks and there are 3 mark scheme points. So any two of those would have got you your 2 out of 2. Now I'm coming to question number 2. B lymphocytes are activated to form plasma cells during immune responses. Figure 2.1 is a drawing of a plasma cell made from a transmission electron micrograph. So we have different organelles labeled. You can see S is the Golgi, R is the rough endoplasmic reticulum, Q is the mitochondria, and P is the nucleolus. Now something is happening at T. They want you to name the process that is occurring at T. Now that is very easy. It is exocytosis, exit. When something leaves the cell, then that is exocytosis. Then it says complete table 2.1 to show the names and functions of the cell structures labeled P, Q, R, and S in the plasma cell shown in figure 2.1. Now, as you all know, P is nucleolus, Q is the mitochondrion, now please remember name. In the name, you cannot give me abbreviations. So P is the nucleolus, Q is the mitochondrion, R is the rough endoplasmic reticulum. Now here you cannot write RER. That's not been allowed. It's a reject. RER or ribosome. So you cannot write RER because it's a name. Somebody just calls you a nickname. That's not your actual name. So please do not write RER because that is, it says it's a name. RER would be the abbreviation for it, which is not allowed. It's a reject in the mark schemes. And S is the Golgi body or the Golgi complex that is allowed, or you can even say the Golgi apparatus. Now, what are the functions of the cell structures in the plasma cells? So, functions of the cell structures. Now, what is the function of the nucleolus? Is the production of ribosomes. What is the function of the mitochondria? Is aerobic respiration, or you can say provides ATP. So, basically, it's aerobic respiration. And then the rough endoplasmic reticulum, what is the function of that? It's protein synthesis. And Golgi body, what is the function of the Golgi body? Packaging of proteins or packaging of the antibodies, or even you can say formation of vesicles or formation of lysosomes. So nucleolus produce ribosomes, mitochondria, aerobic respiration, or you can say provide or produce ATP, and rough endoplasmic reticulum is protein synthesis. And Golgi body, you can say packaging, or you could have even said modification of the protein or the antibody. So packaging of protein into vesicles or modification of the protein. Could have also said that. You could have also said formation of lysosomes. Now coming to the B part of the question, plasma cells can be used in the commercial production of some monoclonal antibodies. This method of production is known as the hybridoma method. Outline the steps in the production of monoclonal antibodies by the hybridoma method. What do? You take the antigen and you inject it into a small mammal, into a small mouse. The immune response occurs in that mouse. Then the plasma B cells are the B lymphocytes, or the plasma cells are extracted from the spleen. Then the plasma cells are fused with the myeloma cells or the cancer cells, and this forms a hybridoma. And then the hybridoma cells are separated to produce clones, and then they will be producing the desired antibodies. And these are called MABs, monoclonal antibodies, on a large scale production. So small molecule injected with antigen, immune response occurs, plasma cells extracted from the spleen, plasma cells fused with myeloma cells to form hybridomas, hybridoma cells separated and used for large-scale production of monoclonal antibodies. Part C, antibodies can be collected from human blood donors and used to treat people that may have been affected with a pathogen. This prevents them becoming ill with the disease. Explain why the treatment does not prevent people becoming ill if they are infected again with the same pathogen. Well, first of all, what is this called? When we inject antibody, that's called artificial passive immunity. Artificial passive immunity, like in tetanus, if you fall down on the road, we give you an injection of tetanus, and that is actually containing the antitoxin. And so you will not suffer from the disease because we cannot give you the weakened pathogen. So this is called artificial passive immunity. There's no immune response. We are just taking ready-made antibodies which we produced in the lab, and we are giving that to you in the form of an injection. Antibodies will be broken down and degraded in your body. Your lymphocytes will recognize it that there is some foreign body, and this is not long term. This is only short term. It's very temporary, and no memory cells are produced, and there is no B cell selection, cloning, and expansion, and antibodies are produced, and plasma cells and memory B cells develop. No such thing is happening. So how do we write this? This is artificial passive immunity. There is no immune response made by the person's own lymphocytes. And as soon as we inject these antibodies, the human being, his own lymphocytes are going to realize that they are not their own, so they're going to degrade it or going to break it down. And this is only a very short-term immunity. No memory cells are produced, and no B cell selection or clonal expansion or antibodies are produced. Now, coming to question number 3, which is a killer question because you're going to really find it difficult to navigate this question, especially if you're giving in your first year your AS exam. In the second year, it becomes easier because you've done A2, which is even more difficult than this. So it says a tyrosine kinase receptor, TKR, is a protein complex found in the cell surface membrane of mammalian cells. TKR has two components involved in the process of cell signaling: a receptor for the signaling molecule, which is a ligand, and an enzyme that catalyzes the transfer of a phosphate group from ATP to an intracellular protein. Intracellular protein. Figure 3.1 is a diagram to show how TKR, the tyrosine kinase receptor, is involved in cell signaling. So you can see the signaling molecule, you can see the outside of the cell, you can see the receptor component, you can see this is the phospholipid bilayer, and then there's this enzyme component, and then you can see the ATP, the three phosphates attached to it, and then you can see the ADP here, and then you can see an activated intracellular protein, and this is the outside of the cell, and this is of course the inside of the cell. This side here is the inside of the cell, and you can see the signaling molecule binding to the receptor component. Shape is complementary to it. It says most enzymes are specific to one reaction. With reference to figure 3.1, explain how the structure of an enzyme provides its specificity. Well, naturally, it's very simple. Enzymes have an active site, and the shape of the active site is complementary to the substrate. So there's one mark for the shape of the active site. Now, some of you will write shape of the enzyme. Now, that will not get you any marks. So shape of the active site is complementary to the substrate or complementary to the intracellular protein. So that will get you your 2 out of 2. Now, shape of the active site is very important that you use this word, and the word complementary is underlined. So that means you had to say, if you said the shape of the active site is is allowing the substrate to fit, well, I'm not going to give you a mark because if you didn't use the word complementary to the substrate, you're not going to get a mark. Now coming to the next part of the question, the drug GN5 is used in the treatment of some cancers. GNF5 affects the activity of TKR by binding to the enzyme component of the complex. Researchers investigated the effect of GNF5 on the activity of TKR using different concentrations of ATP solution. In an experiment, the activity of TKR was measured with no GN5 and with GNF5. Well, I'm not saying GNF, but I'm just saying GN5. The results are shown in figure 3.2. Here we have with no GNF and with GNF. So this is with GNF, and then we have this with no GNF. So we've got two graphs, and you can easily understand it's an enzyme graph, so it's got to be something to do with inhibitors and all that. So this is the activity of TKR in arbitrary units 1, 2, 3, 4, 5, and this is the concentration of ATP, which would be the substrate. And it says the researchers concluded that GNF5 acts as an inhibitor of the enzyme component of TKR and that it is a competitive inhibitor. Use figure 3.2 to provide evidence for these conclusions. Now you look at the graph and you look at lower concentrations of ATP, there is lower rate of activity. As substrate concentration increases, as we are increasing this substrate concentration, the effect of GNF5 decreases. Maximum rate of reaction Vmax is the same, and the Michaelis-Menten constant is higher with the inhibitor. So these were all the 4 points in which you had to give me any three to get your 3 out of 3. So at lower concentration of ATP, there's low rate of activity with GNF5. As substrate concentration increases, effect of GNF5 decreases. Vmax is the same, and Km is higher with inhibitor. Now you can see I've done the Km of both of them with GNF5 and with no GNF5. So the Km is higher with the inhibitor. Now, the last part of this question, a mutation of the gene coding for TKR results in changes to the enzyme component of TKR. This altered form of TKR is known as T315L. The effect of GN5 on the activity of 315 is also investigated. The results of this investigation are shown in figure 3.3. You must read the graph very carefully. Here is the activity of that enzyme, the same thing, the TKR, which is now called T315L, which is now a mutated form. And then here again we have the substrate concentration. And here you can see, I mean, it's some sort of a non-competitive inhibitor because you see the Vmax is not reaching the same. So with no GNF5 and with GNF5. So you have to realize which part of the syllabus are they checking. It's about inhibitors, enzymes. So using figure 3.2 and figure 3.3. This one is 3.3, the previous one was 3.2. State how the activity of T315L differs from TKR when no GN5 was present. So no GN5 is this one. No GN5 is this one. No GN5, this comes here. And similarly here also you're going to look at the no GN5. No GN5 is this one. So no GN5 and then this one no GN5. So you've got to compare these two. And it says state how the activity of T differs from TKR with no GN5 was present. Now, it's only 1 mark, and they expect you to give an answer to that. Now, no activity at 0.1 micromoles. In this, of course, you see what you have to see is at 0.1 micromole, how it differs from TKR with no GN5. So, no activity at 0.1 micromole. You can see here at 0.1 microvolt there is zero activity. So no activity at 0.1 micromole, and maximum rate of reaction Vmax is reached at a higher concentration of ATP. So this is reached here at a higher concentration of ATP, while here this was reached here at a different ATP. So no activity at 0.1, maximum rate of reaction is reached at a higher concentration of ATP, and the Michaelis-Menten constant Km is higher for T315L. Now I've given you the Menten constant. Km is 9 in this one, and it is zero point 0.5 in this one. No activity at zero point 0.1 micromole dm cubed. Vmax reached at higher concentration of ATP, and Km is higher for T315L. The last part of the question, state how the effect of GN5 on T315 differs from the effect of GN5 on TKR. So you have to see effect of GN5. You have to compare now the two graphs of figure 3.2 and figure 3.3. So activity does not reach maximum, does not reach the Vmax. This we are talking about with inhibitor activity of T315L. So please look at the graph. You must have a printed version of the paper and you must be doing it so that you can see what am I comparing. So activity does not reach maximum rate, the Vmax. What are we talking about? We're talking about with inhibitor activity of T315L. And the other is that GN5 acts as a non-competitive inhibitor. And the third point is that GN5 has less of an effect at low concentration on the activity of the enzyme. So with the inhibitor, the activity of T315L, this is the heading. Please remember, this is the heading which I'm talking about. So this is not the answer to the mark scheme. Activity does not reach the Vmax, and GNF5 acts as a non-competitive inhibitor, and GNF5 has a less effect at low concentrations of activity of T315L. So please look at the graphs again and look at this comparison and try to understand how you're going to answer these questions. This was a little difficult question. I agree if you find this was difficult. But please remember, you have to be sensible in looking at the graphs and then comparing them. This was a total 8 marks, and I know this was a difficult question. Okay, now that finishes this video, and we'll continue this video in the second, we'll continue this paper in the second video on this paper. Thank you very much.